Good afternoon. Before we start, the second quiz went up last night, so you can go ahead and take it. The first one is still up. I think it's up through tomorrow. So try and keep up with them, because I want you to have to go back to your notes and remind yourself of what we talked about each week. And if you do, you should be able to get all the questions. They're right from the slides that I give you. So today we're going to talk about virus structure. And there's a lovely quote here from David Baltimore. A virologist is among the luckiest biologists because he can see into his chosen pet down to the details of all its molecules. And virologists are very fortunate in that they have a lot of structures of viruses, and that is going to lead our discussion today. I brought a few viruses with me today, which you've probably seen, these giant microbes, right? You can buy these things. Very smart company. This happens to be polio. And they always put eyes and mouths on these things. At least they make people aware. This is not bad in terms of the structure, as you will see. It's relatively correct. But I have another one here. I wonder if you can guess what this is. Right, HIV, which is totally wrong. This is completely wrong. So we'll see by the end of today whether you can see why. It's got eyes too. Of course, they all have eyes. I don't know why. So viruses are built to be functional and beautiful as well. And that's, of course, the motto of the Bauhaus movement. To create something that functions properly, in essence, has to be explored. It should serve its purpose to perfection. It should be durable, inexpensive, and beautiful. So all of that applies to viruses. They're cheap, durable, and beautiful. So the virion proteins that make up virus particles have several functions. They protect the genome. So inside these virions, or viral genomes, one of the main functions of the capsid proteins is to protect those genomes. So they have to form a stable protective shell. They have to recognize the nucleic acid specifically. You don't want cellular nucleic acids going in there. And in some viruses that have membranes around them, they have to interact with those membranes. When the viruses infect the cell, they have to deliver the genome to the cell. They have to bind receptors, as we'll see next time. They have to uncoat. They have to open up in some way to release the genome. Sometimes they have to induce fusion with membranes. If the virus has a membrane around it, that has to fuse with the host cell membrane. And the virus has to move to the right place in the cell, and that's different depending on what kind of virus it is. It could be the cytoplasm, it could be the nucleus, and the structural proteins are involved in the transport to those sites. Other kinds of interactions besides these with the host throughout the infectious cycle, capsid proteins, structural proteins interact with other cell proteins so that replication proceeds efficiently. We'll talk about some examples of that. Transport to sites of assembly. Now we're talking about the other side of the infectious cycle. Everything has to assemble at the right place. And then once the viruses are released, in an animal, for example, it has to interact with the immune system. Now, one thing that's very important to realize is that viruses are not inert structures. They're not just particles that are passively taken up by the cell. They are metastable structures. And what we mean by that, I'm going to explore in a couple of slides. They have not attained their minimum free energy conformation. Minimum free energy conformation for a virus is gained only when an unfavorable energy barrier is surmounted. And that usually happens during attachment and entry of the particle. The viruses go over this energy barrier. They attain their minimum free energy conformation, and they open up. So the particle itself is not the minimum free energy conformation. This will become clear in a moment. What I want you to think about is that viruses are actually machines, molecular machines that play active roles in the delivery of the genome into the cell. So let's explore metastability a little bit. It's the ability of a non-equilibrium state to persist for a long time. So viruses are non-equilibrium states, or in a non-equilibrium state, and they can persist in that state for a very long time until they reach the right trigger. And metastability is usually a consequence of a very slow phase transformation. So you could keep a virus somewhere for eons if you could. Eventually it would reach its minimum free energy conformation. An example is diamonds. At room temperature, they're metastable because they transform very slowly to graphite. But if you heat them up, we increase the rate of this transformation, and the diamond goes to graphite. So viruses are the same way. We have to give them a way to go quickly through the phase transformation, and that occurs during entry of the particles into cells. Besides the viral capsid proteins, which are metastable, the bonds in DNA and RNA and protein are also metastable. And again, for viruses, this free energy state is gained when we go over a high energy barrier during attachment and entry. So, in other words, to be infectious, the particle has to be metastable. It has to protect the genome. It has to be stable, but it must also come apart. So it has to be both. So we call it metastable. Sort of a paradox, but it's accommodated by the physical properties of the virion. Again, the virus is a molecular machine. It has moving parts, and it does work, as you will see. And we'll see later when we talk about assembly, viruses are actually spring-loaded to uncoat the genome. But they do so only under the right signals. They don't want to release the genome when they're floating around in the atmosphere or in your digestive tract. They want to release the genome upon the proper signal. And that's the same signal that gets the virion over this energy barrier. So when we assemble virions, we put energy into the structure. And when we uncoat, that energy is used for disassembly. So they're active containers. They're not passive. In some cases, actually, replication and transcription of the genome occurs within the particle. We'll see some examples of that. Many viruses open up and the genome comes out, but some, the replication takes place within it, so they are actually containers for the whole process. And often the capsid does not completely come apart. I don't want you to get the idea that one of these viruses gets into a cell and all the pieces fall off and the virion RNA or DNA is left. For many of them, just openings come apart and let the genome out. For example, retroviruses, the RNA is converted to DNA inside the particle. And we'll see for reoviruses, double-stranded RNA viruses, the replication and mRNA synthesis takes place inside the virion. So these are just examples of how the virus particles can also be containers for the different stages in replication. So we're going to talk about the structure of the virion today, the structure of the particle, which is created by the symmetrical arrangement of very identical proteins to provide maximal contact and non-covalent bonding. So this is very important, symmetry and non-covalent bonding. Non-covalency allows you to take apart the virions to let the genome out. The particle has to deliver the genome, so that's one of its other functions. Structure is to maintain the genome and then to deliver it into cells. And this is made possible because we don't have covalent bonds holding all the proteins together. So this is an amazing accomplishment to make a particle like these without covalent bonds. And that's done by taking one or a few proteins and repeating them and having those proteins interact really well with one another. Okay, so some definitions to make sure you understand some of the words that I'll be using. The subunit, I'll say, is a single folded polypeptide chain. So I'll say the virus is made up of subunits, and all I talk about there are the single proteins. Sometimes I'll talk about a structural unit, which is an assembly. It's the unit from which capsids or nucleocapsids are built, one or more subunits, so one or more proteins. The capsid, Latin for box, is the protein shell surrounding the genome. So this gray poliovirus here, this is a capsid. Within it is nucleic acid. A nucleocapsid is the core. Now, the nucleocapsid is within HIV. This is supposed to have a membrane around it. But this is showing an icosahedral capsid, as you'll see in a moment. So the nucleocapsid is the core that contains nucleic acid. It's typically protein mixed with nucleic acid. Envelope is another name for the viral membrane, and that's derived from the host cell. Host-derived lipid bilayer. The virion, of course, is the infectious viral particle. When I say virus, I'm using it as a generic term. Virion means a very specific thing. This is a virion, but only if it's infectious. If it's not infectious, which many viruses are, it's not a virion. So infectivity is inherent in the definition. So to put the size into perspective, again, we touched on a little bit of this earlier. A nanometer, of course, is 10 to the minus 9th meter, so atoms are about 0.2 to 0.3 nanometers in diameter. An alpha helix in a protein, about a nanometer in diameter. DNA 2, a ribosome 20, and poliovirus 30. Remember, we showed a drawing of ribosomes next to polio, similar sizes. And the very large mimiviruses, 750 nanometers. Here's some more size comparisons. This is an illustration of proteins that are in the protein data bank, and these are based on three-dimensional structures. Here you can see a rhinovirus about the same size as polio, which is the gray virus sitting up there. And these are all other proteins that you would find in a cell of various sorts, various enzymes. Here's trypsin, for example, synthetase, and so forth. So you can see viruses are starting slightly larger than most cell proteins and get very, very much larger. So first we'll start with this very important experiment from 1955 at UC Berkeley, the first deconstruction and reconstitution of a virion. What they did was to take tobacco mosaic virus, which is this rod-shaped virus here, and they broke it into its protein and RNA components. You remember that was the first virus discovered. It was also the first one from crystals, of which crystals were made, and the first one to be shown that RNA is the genetic material. When they took the protein and the RNA that they had separated and mixed them together, virus particles formed spontaneously. And those virus particles were infectious. So this is showing that all the information to assemble a particle is in those two components. Now, the structures that we're going to talk about today were determined by a couple of techniques, and these are the tools of viral structural biology that are used today. These are described in some detail in Chapter 3. Electron microscopy, X-ray crystallography, cryo-electron microscopy, and nuclear magnetic resonance spectroscopy. We'll go over some of these. So what we know about the structure of viruses really began in 1940 when Helmuth Ruska used an electron microscope to take the first pictures of virus particles. And there's the article in German which reported that finding. And electron microscopy depends on getting contrast into biological materials which inherently have little contrast. If you've worked with any kind of cell, or tissue sections, there's very little contrast in that. So they have to be stained, but they can't use just any stain to be examined by electron microscopy. You have to use an electron-dense material because, after all, you're bombarding the specimen with electrons, and it will pass through most things except stains that are refractory to it. And these are some stains, uranyl acetate, phosphotungstate, that will scatter electrons when the electrons hit the stain. So you stain with those materials. The resolution is not very good. So the smallest structures you can see in electron micrographs are about 50 to 75 angstroms. And just by contrast, an alpha helix of a protein is 10 angstroms in diameter. So you cannot see individual protein chains, obviously. So no detailed structural interpretation is possible. And so typically there are two ways you can stain viruses. On the left is shown one way by putting the stain covering the entire particle. So you put the viruses on a grid, shown in orange. You put the stain on top, which is this gray material, and then the electron microscope bombards it with X-rays. And wherever there is an electron-dense material, the X-rays don't get through. The electrons don't get through. And this is a kind of image that you can pick up by doing that. So when you do this, you stain the background, which gives detail to the particle. So this stain is molding around the particle, and it's making an outline of the particle. But when you cover the particle with stain, you get both the top and the bottom at the same time of the particle, so you have a mixed image. And that is often making it difficult to distinguish fine structures. So typically what's done is to only bring the stain up about halfway, as shown here. You get only one part of the particle, and that gives you more accurate structural information. And just to show you the difference, when you cover the particle with stain, you get this kind of image, which probably looks perfectly good to you, but when you only cover half the particle, you then begin to resolve these substructures, these donut-shaped substructures, which are very important. And you don't see those in the other because the two sides of the virion are stained. So here are some very nice images of viruses. We'll just turn the light off for a moment so you can appreciate them. These are all electron micrographs, negatively stained viruses. Herpes virus up here, hepatitis B virus, the famous picture of adenovirus. The capsid is here, and you can see these antennae, if you will, coming out. Influenza virus, a picornavirus. And you can see here some of these are empty. They're broken. They've lost their nucleic acid, so the stain doesn't pass through them. And a pox virus here. So very, very nice pictures. And these, you can imagine when this was first done, people immediately saw what viruses looked like. They'd never known that before. All they knew was that they were something that could pass through a filter and infect various organisms. So that's electron microscopy. Now you take a variant of electron microscopy and you can do much higher resolution structural studies, and that's cryo-electron microscopy, which will give you resolution between 8 and 20 angstroms. In fact, now it's getting even lower in some hands. What you do here is you take a preparation of virus particles. Here's an electron micrograph. And you don't stain them, you just freeze them. And the freezing gives them just enough contrast that you can see them in the electron microscope. And then you take pictures of the individual particles. The assumption here is that every particle is oriented slightly differently on the grid. So it's basically like doing a CAT scan of a person where you rotate the X-ray machine around them and get multiple slices. You're doing the same thing. You take a few hundred pictures of viruses and you digitize the images, and then you use Fourier transformations to convert the images into phases, and then you mix them all together. So here we're showing 1, 2, 3 different slices, but in effect you would have hundreds of slices, and then you do an inverse transformation to make the particle from that. So you're basically taking slices of virus images in many different orientations and using software to reconstruct the entire virion. So that's why this is called three-dimensional reconstruction, because you're reconstructing it from a two-dimensional image. So that's cryo-electron microscopy. This is the kind of image you can make with that technique. This is poliovirus, okay, the same virus on gray up here in the desk. It's now shown in red. And in this case, the virus was mixed with its cellular receptor, a soluble form of the receptor, that is, without the cell. And those are in green. And so you can see very nicely, you can resolve the virus particle as well as the individual receptors binding to the surface of the virus. Now, the highest resolution comes from X-ray crystallography. In this technique, you have to make a crystal. You have to take your purified virus and find conditions under which it forms a very nice crystal, a lattice of molecules. You bombard the crystal with X-rays, and then you'd make an image of the scatter of the X-rays. In the old days, it was done on film. Now it's done on an image sensor. And the idea is that as you bombard this with X-rays, the X-rays bounce off different atoms in the crystal and end up on the scanner, and they are a representation of the crystal lattice. And so you turn the crystal in various orientations and bombard it, and eventually you can deduce the three-dimensional structure by phase transformation, similar to what was done for cryo-EM. The resolution here is much greater. It can be less than 2 angstrom in many cases. So the genesis of structure biology began in 1935 when Stanley crystallized tobacco mosaic virus, another first with that virus. He just made crystals, and he showed that if you dissolved the crystals, then the virus was still infectious. So that meant it was being crystallized in some biologically relevant form. And for that, he got the Nobel Prize in 1946. But that wasn't the first virus whose structure was determined, unfortunately. It was this one, tomato bushy stunt virus, TBSV. This was solved by X-ray crystallography in 1978. And it's not really red, of course, but I suppose they took some license from the name. This is a plant virus. It infects tomatoes. The first animal virus whose structure was determined as poliovirus. These are two different images showing you the resolution of X-ray crystallography on the upper left. You can see all the individual amino acid chains, and cryo-electron microscopy. This is about 8 angstroms. So you can't see individual chains, but you can see the overall morphology of the particle. Now, how do you build viruses? Symmetry is the key. And this comes from the work of Watson and Crick, which you may remember were two individuals who proposed the original structure for DNA. They also had a big role in our understanding of how virions are built. So in 1956, they published a paper saying most of the virus particles that we've looked at under electron microscopy by that time were either spherical or rod-shaped. You didn't see anything else. And they also knew that particles were made with many copies of a few proteins. So up to that point, some biochemistry had been done on virus particles. And for the most part, there were just a few proteins, and they seemed to be repeated over and over to make the virions. They also deduced that viral proteins have properties that allow regular and repetitive interactions among them. This is very important. This is not something we find in the world around us except in viruses, that you have proteins that are built that can repeat with one another over and over in the same way. So their contribution to virology was the conclusion that identical protein subunits are distributed in the virions with either helical symmetry for rod-shaped viruses or platonic polyhedrous symmetry for round viruses. So a rod-shaped virus is like tobacco mosaic virus, the long rod shapes. These are round viruses, okay? That's what they meant by that. That was their contribution. There are two general ways to build virus particles. This is pretty much correct even today. There are some variations on these themes, but they were quite right. It's as important as was the discovery of DNA, I think. So the symmetry rules provide the rules for self-assembly of particles. And here they are. Rule 1, each subunit, remember, a subunit is a protein. Each subunit has identical bonding contacts with its neighbors. This isn't quite correct, but it's close enough for this point in the story. The repeated interaction of complementary surfaces at subunit interface naturally leads to a symmetric arrangement. So again, the proteins, the subunits of a virus are repeated. They interact with each other in very similar ways. That gives you a symmetrical arrangement. Rule 2, the bonding contacts are usually non-covalent. And again, that's important because you have to take these particles apart. If these virions were made of covalently linked proteins, they would never get apart upon infection. The reversible formation of non-covalent bonds between properly folded subunits leads naturally to error-free assembly and minimizes the free energy. So regular structures form when identical bonds are made between identical subunits. You take a protein, and we'll illustrate this in a number of ways. If you make identical bonds among the subunits, you get a regular structure. And that's unusual because if you make non-identical bonds between different proteins, you get aggregates or clumps or disordered complexes. And that is the unique aspect of virus assembly. So we have repetitive interactions among a limited number of proteins. That gives you a regular structure. And in fact, if you express just the capsid proteins of a number of viruses, they will self-assemble into capsids. You can take some viruses, there is one protein that you can express and it will make a virus particle. And a great example are the vaccines for hepatitis B virus and human papillomavirus. These are made by expressing one or a few proteins of the virus in yeast or in other cell systems. For the papilloma vaccines, it's just one protein. It's expressed and they make particles, and these are shown here in the electron micrograph. So all the information needed to make a particle is in the primary sequence of the protein, and you get repetitive interactions between the proteins, and they form a capsid. Now, just on this past week in our podcast, TWIV, we got a letter from Sheldon in Toronto, and he says, please explain how virus-like particles are created for Gardasil and Cervarix and those being developed for flu vaccines. It seems somehow magical that you can build something that is structurally a virus on the outside, but is essentially an empty shell. So he's absolutely right. It's magic because it's in the nature of the protein. So I just wanted you to see that there are people out there wondering how this works. Now you know, because the proteins have this ability to interact with one another in symmetrical ways. That's why you can get these virus-like particles. So let's explore the two types of symmetry. The first is helical symmetry. It is the way tobacco mosaic virus is built, and also a number of viruses that infect people. And what happens here is that we have a coat protein, and these engage in very similar interactions with one another, and also with the viral RNA, which is shown here in in green. All right, so each of these is the coat protein subunit. The tobacco mosaic virus is just made of one protein. It's repeated over and over, and it forms this helical structure. Okay, and so that is the virus itself, the protein and the RNA. It doesn't have anything else associated with it. These are some examples of animal viruses that have helical symmetry. This is Sindbis, the nucleocapsid. So we call this the nucleocapsid now because it is part of a bigger structure. This is usually surrounded by an envelope in animal viruses. And this is vesicular stomatitis virus or rabies virus. They both look like this. Same kind of symmetry. A coat protein wrapped around in a helix with the nucleic acid. And here is tobacco mosaic virus. So that's the virus particle, just this rod-like structure composed of the protein and the RNA. And this is vesicular stomatitis virus. The helical nucleocapsid is in here, and it's wrapped with an envelope. This is not really a very good depiction of this. Okay, it's not really a coil. It is a very tight structure that looks just like that. And this is Sindbis virus. These are the nucleocapsids. That's this guy, this long rod right here, and they're encased in an envelope, and this one is broken, so the nucleocapsids are spilling out. But you can see how they would normally be contained within the envelope. Now, you can make your own demonstration of the helical symmetry with these buckyballs that you can buy. Okay, you can go, in fact, Saul Silverstein gave me these a year ago, and I said, I can build a virus from this, and it's what I want to show you. So these are very strong magnets. No, it did not. No, actually, okay, so this is helical symmetry, okay? And each of these magnets is a protein subunit. It interacts with each of its neighbors in an identical way, and it makes the capsid of that virus that was on the screen before. So the RNA is attached to this. You don't have RNA here, but you can see there's a helical rise here. So it's just the way a virus is built. Perfect. It didn't take me a year. I had these on my desk, and one of my students was playing with it, and she assembled it into a long strand, and I said, I'll bet you could make that into a helical nucleocapsid, and here we go. So I've been waiting to show you this for about a year. Okay? So that's pretty cool. Those are buckyballs. I have a movie here for the people who are going to watch online who aren't here, so they can see it as well. It's basically me doing the same thing, wrapping up this chain of buckyballs and making a virus out of it. Now, the interesting question, yes?
Yeah, so the nucleic acid bonds spiral into protein.
So each subunit also binds the nucleic acid.
Okay, but it's within the same way.
Yes. Now, that's a good question, because if I took all of these magnets and threw them in a cup, they wouldn't form a helix. They need the RNA to do that, the nucleic acid, because that drives their assembly into this kind of structure. So this is not a perfect example of what helical symmetry is because there's no RNA involved, but you get the idea, basically. Okay. That's it. I got to put that on YouTube, see how many hits it gets, yes?
So relating to that person
Yeah.
who emailed you that question,
Yeah.
so how do they assemble them? They just throw the proteins together and they automatically
No, they deliver the gene encoding the protein into a cell, either a yeast cell or an insect cell, and the gene is transcribed to mRNA. It's translated into protein within the cell, and then the proteins self-assemble. So you could take the purified protein in a tube, and it would also self-assemble, most likely. But for production purposes, for making a vaccine, you need a lot of quantity. They express it in cells. Okay. Yes?
So with these helical shells that each bind the nucleic acid,
Yes.
is it possible to assemble just the protein without the nucleic acid to make a
So the virus-like particles that we talked about are made by symmetric, they're icosahedral structures. I don't know of any where a helical structure has been made with just the protein. It's my understanding that you need the nucleic acid to nucleate the helix, right? Was there another one? Okay. Yes?
So in the virion, are the nucleic acids, are they like held onto the capsid by covalent forces?
Well, in these helical structures, they are, yes. But in the capsids, these icosahedral capsids that we'll talk about next, we don't know because we assume there are some interactions, but we haven't been able to visualize them structurally because the RNA or the DNA is not ordered inside the capsid, so it's hard to see the interactions it's making. But when you solve the structure of these helical nucleocapsids, you can see the nucleic acid interacting with the protein because it's given order by the protein order. Okay. So this is just an example of this interaction. So here's a virion of vesicular stomatitis virus. The nucleocapsid, the helical nucleocapsid is within a membrane. So again, here's the same image I showed you before, which isn't quite right because it's not a coil. This should be much tighter. And this is made up of RNA plus the N protein, nucleocapsid protein. It's a single protein. The structure is shown here. And here is an image of 9 of those. And there's a green molecule snaking through it, which is the RNA. So you can see the structure of how the RNA interacts with the protein. So this is actually data from X-ray crystallography. So and here on the right, you can see 1 N subunit and 9 nucleotides of RNA, how they bind it. And when you put many of those together, they form a helix. Okay, so the interaction of the protein and the RNA is shown here. For all the animal viruses I know of that have helical symmetry, they all have envelopes. None of them are naked like tobacco mosaic virus. Okay? I don't know why that is. It may be related to the way these viruses are transmitted in nature. So here are just some examples of envelope virions of RNA viruses. These are negative strand RNAs with helical capsids. Measles virus, Paramyxoviridae, rhabdo, rabies virus. Influenza viruses have this helical structure, Ebola. And for all of these, the capsids are always called nucleocapsids because they're within a membrane envelope. Again, the nucleocapsid, the nucleic acid protein assembly that is packaged within the virion. The nucleocapsid is not the virion for these viruses. For tobacco mosaic virus, we call it the capsid because that's it. There's nothing else. Okay? That's not really so important. Just I'm telling you this so you don't get confused in the future. All right, now moving on to round viruses. Remember, these are the round viruses, these guys like this. How do you make a round capsid from proteins with irregular shapes? Now, the magnets are not really instructive because they're all the same shape. And in fact, these proteins are asymmetrical. The ones that form these helical nucleocapsids are asymmetrical. But again, it still illustrates the principle. But even harder is how to make a round capsid from these different proteins. And the first clue is that all round capsids have precise numbers of proteins, always. And they're usually multiples of 60, although that can vary. But for the most part, they're multiples of 60, 60, 180, 249, 60, etc. So that gives us one clue. Next, spherical viruses come in many sizes, but capsid proteins are usually between 20 and 60,000 Daltons in size. So you can get very big capsids. Remember, Mimi virus is 750 nanometers, but the proteins aren't that big. So somehow the two have to come together. All right, so Casper and Klug, two structural virologists in 1962, solved this issue. They knew from Watson and Crick that round capsids are also icosahedrons. So remember I told you originally they were a platonic solid, but in fact there are many platonic solids, and only one is ever used to build viruses, and that's the icosahedron. None of the other ones. They also observed that capsid subunits tend to be arranged in groups of 6 and groups of 5, hexamers and pentamers. And that the number of capsid subunits in the virions followed allowable values of T numbers. All right, we're going to talk about a T number in a moment. 60, 180, 240, 960, and nothing in between. All right, so those were the facts they started with, and they said icosahedral symmetry turns out to be the most economical way to build a symmetric shell with maximum internal volume using non-symmetric proteins. Okay? Of all the platonic solids, this is the best one that fits all the bill. Symmetric shell, maximal internal volume, because you want to put a genome in there, and using proteins that are not spheres, but they're asymmetrical. That's the icosahedron. Icosahedron, of course, is a solid with 20 faces, each an equilateral triangle, has 12 vertices, and what are called axes of rotational symmetry. That'll be more obvious in the picture in a moment. This allows the formation of a closed shell with the smallest number of subunits. So that's the smallest shell you could make, one of 60 subunits. Can't make anything smaller. We don't know any virus that has fewer than 60 subunits. We do know viruses with more. I'll show you how to build those as well. But an icosahedron allows you to do this because of the geometry. So here's a drawing of an icosahedron. Remember, it has 20 faces and 12 vertices and 5, 3, 2 axes of rotational symmetry. So 20 faces, this is a face, these are the vertices, and these are the axes of symmetry, a 5-fold, a 3-fold, and a 2-fold axis. And all that simply means is that there are 5 copies of a subunit around it. So 5-fold axis, in this one there are 12 faces around it. If these are made of one protein, there would be 5 proteins around the 5-fold axis. Same for the 3-fold and 2-fold. They're mostly used for reference points. So sometimes I'll point out a virus picture and I'll say the receptor binds around the 5-fold axis. I just want you to know what I'm talking about. That's with reference to this. So here are a few other views of these 3-fold. Looking down at the 3-fold axis, it's the same as that. We're looking now down at the 5-fold axis, so 1, 2, 3, 4, 5 subunits around it. And here's the 2-fold axis. There are 2 subunits. So here is a drawing of a simple icosahedral capsid. It's made of 60 subunits. Remember, that's the smallest virion we can make using this symmetry. The protein subunit, shown as a comma here, is the structural unit. And all these interactions, the interactions of all these commas, again, each comma is a single protein. There are 60 of these in the virus particle. Their interactions are all the same in the particle. So every protein interacts with its neighbor in the same way. Head to head, tail to tail. All right? So here's the 5-fold icosahedral axis. You can see 1, 2, 3, 4, 5 copies of the protein around it. 2-fold icosahedral axis, 1, 2 copies, 3-fold, 1, 2, 3. Now you also see there are some areas where there are 6, 1, 2, 3, 4, 5, 6 copies. So in some of these, in some of the viruses, you do have 6-fold interactions as well. So that's the simplest capsid you could build. 60 subunits. Proteins are asymmetrical, and that's the point of drawing it as a comma. You have the head and the tail, but they all interact with each other in very similar ways. So here's an example of such a virus, a parvovirus, the kind of virus that can infect your dog or cat. It is a very small capsid, 25 nanometers in diameter. It's made of 60 copies of a single capsid protein. One protein repeated 60 times. It's an asymmetrical protein, but it can interact in ways very similarly throughout the capsid. And that's what the protein looks like here, and that's the virion. So, for example, these different parts of this capsid protein are colored. So the blue part here you can see forms the 5-fold axis of symmetry. It's very easy to find it. You can just look at the point where there are 5 repeated subunits around it. 1, 2, 3, 4, 5. So all the blue is contributed from 5 different proteins, and it's this specific part of the protein. So how do you make a bigger virus? So you have 60 copies of a single protein here. How do you make bigger viruses? You just add more subunits. Very simple. And this relates to the triangulation number T. T is simply the number of facets per triangular face of an icosahedron. So here's a triangular face. Has 1 facet, but you could take that triangular face and insert 3 additional facets into it. It gets bigger. This is still the triangular face, but now we have facets in it. And that's achieved just by adding more proteins to it. And if you want to make bigger viruses, and we know that they exist, we're trying to explain how they are made, you simply add facets by adding proteins. So this is called a T=1 virus, and this is called a T=4 viruses. And any capsid with a greater than T=1 T number will have a 6-fold axis of symmetry. So I misspoke in the previous slide. That T=1 particle does not have a 6-fold axis. It has all 5-fold, 3-fold, and 2-fold. But this is now a T=4 virus. You can see 1, 2, 3, 4, 5, 6, in addition to 3, 2, and 5-fold axes. So simply putting in more proteins to make a bigger virus now gives you not only pentamers, or 5-fold axes, but also 6-fold axes as well. Okay, the T number simply refers to the number of facets per triangular face. Here we have 1, 2, 3, 4 facets. The T number is 4. Here we have 1 facet in the triangular face. The T number is 1. So you'll see T number in the book and in other materials that you read. I just want you to understand what we're talking about. You can use the T number to calculate the total number of subunits in a virion. So it's 60 times T always. And again, the simplest virus is T=1, 60 subunits, 1 virus particle. So for example, a T=3 icosahedron has 180 subunits, 60 times 3, and this is in sets of 3. T=4, 240. Now, not all viruses follow this T rule, 60T. And you don't need to really understand why, but on this table are some examples of that. Here, for example, this simple parvovirus, the number of subunits, or 60T, is actually 60, and that's what we observe. But you can see that for simian virus 40, 60T is 420, but there are only 360 copies of one of the proteins. So not all the rules follow, but they're pretty good overall. All you have to know is that some viruses do not contain 60T subunits, and you don't have to know why. Now, when we put more when we put more proteins in a capsid to make a bigger particle, to go from this to something much bigger, you get more than 60 subunits. We increase the T number. Now we encounter something called quasi-equivalence. Remember, when we had a small virus with 60 subunits, all the subunits, all the proteins interacted with each other in identical ways. When you start putting more proteins in to make bigger capsid, that can't happen anymore. They're pretty similar, but not exactly the same. So we call them quasi-equivalent. Okay? That's all that means. When you put more than 60 proteins in a particle, each subunit or each protein occupies a quasi-equivalent position. What that means is that the non-covalent binding properties, they're always non-covalent, of subunits in different structural environments are similar but not identical. So only in the T=1 60 case are the interactions all identical. Anything bigger, they're not. So this is an example of a T=3. Now we've put in extra subunits to make the capsid bigger. Now we have 3 modes of subunit packing. We have orange modes. Orange modes here. We have yellow modes. And we have purple modes of packing. We have both pentamers and hexamers. Now, the hexamer is something you get, remember, when you add more than 60 protein subunits. And the bonding interactions are quasi-equivalent. All of these proteins engage in tail-to-tail and head-to-head, but they're slightly different depending on where they are in the particle. So that's all that quasi-equivalence means, is that the interactions are not quite identical, and that's a consequence of trying to make a bigger particle. So this T=3 virus is bigger than the T=1 that we first started looking at. So here's an example of a T=3 virus. It's called Nodomura virus. This is a virus that infects insects. It's made up of 180 copies of a single capsid protein, and in this image on the lower left you can see the various rotational axes. Here's a 6-fold axis, and here's a 5-fold axis as well. 5-fold axis of symmetry, 6-fold. Again, the 6-fold arises because you're putting in more proteins. It's as simple as that. Now, poliovirus is an example of a T=3 capsid as well. It's made up of actually 4 capsid proteins, but the 4th one is simply an extension of one of them. So in reality, it is a T=3 virus And it's called pseudo T equals 3 because you actually have 4 capsid proteins. Again, you have 5-fold axes of symmetry. Here are 5 copies of VP1 surrounding the 5-fold axis, and there are 6-fold axes as well. Now, what's shown here is to give you an idea of how you actually build a shell from these proteins. All of these proteins that form capsids actually have volume, of course, and that's diagrammed here. So all of the poliovirus capsid proteins can make a wedge-like structure when they're folded properly, and these wedges simply pack together to form the virion. So the thickness of the wedge gives you the thickness of the virion itself, and they make a very nice closed shell because they're wedges and they interact non-covalently. And I think on the next slide we have an illustration of how that happens. So here's a surface view of poliovirus. Here's your 5-fold axis of symmetry. You can see the star up there. Here's a 6-fold axis. And again, this is T equals 3, so you have to have 6-fold axes. And here's a cross-section of the virion. So you can see this is the thickness of the shell which encloses the interior. The RNA would be in here. And that thickness is formed by those wedges. The proteins are wedges that pack together to form this shell. Now, in all of this is a structure determined by X-ray crystallography. In all the structures, we don't see the nucleic acid. That's because it's disordered in the interior. In order to get crystal structures, the proteins have to form an ordered array. And the nucleic acid doesn't do that. So that's why we don't know how the nucleic acid is interacting with the capsid. For a few viruses, there are a few nucleotides visible in the crystal structure. And that gives you a little clue about how the RNA is interacting with the protein. But for the most part, we don't understand how they are packed in. Now, there are even bigger viruses that we can see that are built. These are called structurally complex. They do have, for the most part, icosahedral symmetry, but they are much larger than this, and they have many more components. So they all follow icosahedral symmetry, but they have other proteins inserted as well that have specialized functions. So here's an example of adenovirus. This is not something you would ever have to remember. I'm just showing you this as an illustration. These are all the various proteins that are present in the capsid of adenoviruses. So you have hexons and pentons, which form the icosahedral capsid, but everything else are additional proteins that have specialized functions. For example, there's a fiber protein. If you remember a few slides ago, I showed you an electron micrograph of adenovirus, and I said there are these antennae sticking out. Those are the fibers, and those are what are used to attach to cell receptors. So there are 12 of those because there's one for each 5-fold axis of the icosahedron. And there's a variety of other proteins as well, both in the capsid shell and inside binding the DNA. So that's what we mean by proteins devoted to specialized roles, not just building the shell, but they have other functions as well. And here is an example. So this is adenovirus. Here's the icosahedral shell. And the hexons and pentons are the major proteins that comprise the shell. Then there are the fiber proteins that emanate from each 5-fold axis. Again, those attach to the cell receptors. But then you see these little proteins in between, these brown ones here and green ones underneath. These have other functions. Some of them are thought to glue the capsid proteins together to help them stick together. Of course, they're not glue. They're just non-covalently bonded. But for some large capsids, it's believed that other proteins have to stabilize it. In the middle of the virion, there's the nucleic acid, and that's also bound to proteins as well. So these are very large particles now, 150 nanometers, going up from 20 to 30, the smallest ones. These are T equals 25 now, so 25 times 60 would be the right T number. And there are fibers at 12 vertices. This is the structure of some of the components of this virus particle. Here is the capsid. You can see the nice icosahedral symmetry. Here's a 5-fold axis of symmetry from which the fiber emanates. It's gone in this image. And these are also hexons, which are surrounded by 6 different hexons. So here, the penton is surrounded by 5 hexons. The hexons are surrounded by 6. This is just the structure of the fiber that emanates from the 5-fold vertices. So again, illustrating this idea that you have, in addition to proteins that build the shell, a lot of other proteins in these big viruses that have other functions, binding to receptors, protecting the nucleic acid, gluing the capsid together, and so forth. There are some very interesting viruses that actually consist of 2 icosahedral shells, concentrically arranged, and that's the reoviruses. These are T equals 3 viruses. They have 2 concentric shells, one of 70 nanometers and one of 90. So just imagine an icosahedral shell. We've just built several of them, and there's simply 2, one around each other. And that's very unusual. Now, in some cases, those capsids, the virus is just a capsid. So this is poliovirus. This is an icosahedral capsid. 1, 2, 3, 4, 5. I can count 5-fold axes of symmetry. There are no 6-fold axes on here. I don't know if they couldn't do it or they didn't know, but there should be some 6-fold. Okay? Anyway, this is the virus of polio. That's it. For some, though, the capsid is covered by a membrane. All right? Covers this thing. And that membrane is very common. These membranes are very common in viruses, particularly those that infect animal. There are very few envelope plant viruses. We don't know why. It's derived from the host cell because the virus can't make its own lipid. It doesn't have any machinery to do that. So all envelope viruses derive the lipid from the host cell. It can be acquired by budding through a cellular membrane, and it can be almost every membrane in the cell. It can be a nuclear membrane, can be a plasma membrane, but it always virus specific. So the influenza viruses always bud from the plasma membrane, and the herpes viruses bud from the nuclear membrane. Can also be other membranes in the cell as well. The nucleocapsids in the envelope can have helical or icosahedral symmetry. Okay, so remember the helical symmetry. This kind of symmetry, this can be wrapped in an envelope, and for animal viruses it always is, but for plant viruses it's not. And then we can have these icosahedral particles also wrapped in envelopes. So that gives you other options for structure as well. So if you wrap a virus in an envelope, so take this and you wrap it in an envelope, if you just use a membrane from the host cell, there's nothing specific about that membrane. You have to give it viral properties. So all viruses put proteins in that membrane derived from the host cell. And these are called viral envelope glycoproteins. So the envelope is the membrane surrounding the virus. The viruses put their own glycoproteins in. These are integral membrane glycoproteins. That means there's a part of the protein on the exterior of the virus. Here's the viral membrane. There's a transmembrane segment, and typically some protein sequence in the interior of the virus as well. These have a variety of functions in replication. They're used for attachment. They are used to generate antibodies. When you make antibodies against these viruses, typically these are the proteins that antibodies are directed against. And also when these viruses fuse with the host cell, as we'll see next time, these proteins play an important role. And they're often oligomeric, and they're originally called spikes. So when the first electron micrographs of these viruses were done, here's influenza virus, they were said, oh, these look like spikes on the surface. So that's what the name has stuck since then. But they are each individual viral glycoproteins. The viral glycoproteins can be perpendicular to the membrane, as shown here. On the left for the HA, the hemagglutinin of influenza virus. That's very typical. But other viruses, flaviviruses, West Nile virus, the glycoprotein is horizontal. It's lying down on the membrane. Both ways work, just two distinct topologies. So here's how these horizontal membrane glycoproteins look from the top and from the side. And here's how they would be on an intact virion. So you see the virion surface has these glycoproteins lying down. You may notice here that there is some symmetry here, right? Doesn't this look like a 5-fold axis of symmetry? Well, it turns out that some of this particular virus has a membrane around an icosahedral shell. And the icosahedral symmetry of the shell is being transmitted to the glycoproteins on top. And we're going to talk about that now. So these viral proteins, these glycoproteins, interact in at least 3 ways with the virion. On the left is an example where the glycoprotein is going through the virus membrane and it's interacting with this capsid or nucleocapsid This would be more accurately called a nucleocapsid because it's wrapped in a membrane. But you can see the end parts of the viral glycoprotein are interacting with the capsid protein, so that's one typical arrangement. You can see if that's the case, then the symmetry of the nucleocapsid should be transmitted to the glycoproteins on the surface. Okay, so if this glycoprotein is interacting with one of the 5 proteins around the 5-fold axis, then the glycoprotein will also assume 5-fold axis of symmetry. And that's what was happening in the previous slide. Sometimes there's an additional protein below the membrane of the virus. It's called a matrix or an M protein. It's in blue here. And that contacts the capsid, and in turn it also contacts the viral glycoprotein. Or you can have, some viruses have multi-protein layers, where there's more than 2 layers between the glycoprotein and the capsid of the virus. Just different ways to build particles. Now here's the structure of a togavirus. It's a positive sense RNA virus, single strand. It's called Sindbis virus. And this is an example of how the symmetry of the capsid is transmitted to the glycoprotein. So this is an envelope virus, has glycoproteins in the envelope. Within the envelope is an icosahedral shell. The glycoproteins contact the shell directly. So here is a cross-section of a single virus particle. This is actually an amazing structure. This is the RNA in the center. On the outside are the glycoproteins. And here you can see the lipid bilayer, the 2 leaflets of the membrane. The glycoproteins are passing through the leaflet, the bilayer, and they are contacting the capsid. So here are the capsid proteins here surrounding the RNA, and the glycoproteins are contacting the capsid proteins directly. As a consequence, if you look at the outside of the virus particle, which is shown here, you see 5, 3, and 2-fold axes of symmetry in terms of the glycoprotein arrangement. Right? The icosahedral capsid is on the inside, but because it interacts with the viral glycoproteins in this manner, the glycoproteins assume the symmetry of the underlying capsid. So that's called a structured enveloped viruses, because the capsid underneath gives symmetry to the glycoproteins on top. Many viruses are unstructured. Many envelope viruses are unstructured. And here are 3 examples. Vesicular stomatitis virus, again, it's an envelope virus around a helical nucleocapsid. There's no icosahedral symmetry here. This is helical symmetry, but there aren't contacts between the nucleocapsid and the glycoproteins such that the nucleocapsid would transmit any sort of regularity to the viral glycoproteins. Same with influenza virus. It consists of helical nucleocapsids surrounded by an envelope, but these are not contacted by the viral glycoproteins. Same with retroviruses. So unstructured virus or unstructured envelopes, there's no contact between the interior and the exterior glycoproteins that would give the surface any kind of regularity. Now, herpes viruses are examples of very large viruses that are built on the principles of icosahedral symmetry, but which have also additional viral proteins like adenoviruses. And I find these amazing because these are 2,000 angstrom virions. Over half of the viral genetic information is devoted to building this capsid. So the virus has 80 genes, and over 40 of them encode structural components. So that's a lot of genetic cost to make this shell. So to make a big virus is not cheap in terms of genetics. So there are 13 envelope glycoproteins and 2 distinct internal structures. There's an icosahedral nucleocapsid that surrounds the DNA. This is made up of 4 different proteins. So this is a typical icosahedron. It has a T number. We know the number of subunits of it. It also has what's called a tegument made of 20 different viral proteins, which is surrounding the nucleocapsid, and it's involved in delivering proteins required early in infection. So here is an image of herpes virus solved by cryo-electron microscopy. Here's the intact virion here on the left. The yellow are the glycoproteins on the surface of the virion. And in the interior, we have the icosahedral capsid that's in the light blue here. And then the orange is the tegument. The blue is the membrane. The orange is the tegument. So you can see the It's a typical nucleocapsid, icosahedral nucleocapsid, but has these other structures as well. Now, in addition to this, the icosahedral shell has a very interesting feature, which is it has what's called a portal. And I think we'll turn the lights out to see this portal. You can see the portal here in this electron micrograph. It's right there, and that's what it looks like. There's one portal per virion, and that portal is used to get the DNA in and out of the particle. So it's built at one of the 12 five-fold vertices of the capsid. So the five-fold vertices are in red, and one of those is devoted to being a portal. And that's how we think. Here's the portal here at one of the five-fold axes. You see there's a hole there. None of the other five-fold axes have that hole, and we think that the DNA goes in and out of it. So another example of a specialized structure built on top of icosahedral symmetry. What else is in virions besides all these proteins we've talked about? There are enzymes of various sorts. Some viruses have to bring enzymes into the cell to replicate the genome. You'll see in the retroviruses there are integrases so that the genome can be integrated into the host cell. Enzymes to cut proteins of various sorts. Enzymes to make poly-A tails, to put caps on mRNAs, to unwind double-stranded DNA. Other proteins are needed for the synthesis of mRNA transcription or other parts of the replicative cycle. And even cellular components. Some viruses package histones, tRNAs. There's also lipids of various sorts and all sorts of molecules in virions besides the structural proteins that we've talked about. And I just want to show you two examples of this. This is poliovirus, and this is the structural subunit of the virus, which consists of three different proteins, blue, yellow, and red. The fourth is an extension of the yellow protein. That's why it's pseudo T equals 3. 60 of these are assembled to make the virion. This is the final virion on the bottom, which we've seen before. This molecule in white is a lipid. It's sphingosine, a long-chain lipid, in which there is one per subunit. So since there are 60 subunits, there are 60 copies of this lipid in the final structure, and you can see some of those here around the five-fold axis of symmetry. So the five-fold axis is built right here, and these lipids surround the five-fold axis. We think these lipids have a role in entry of the virus into the cell. We will talk about that next time. In influenza virus, there's another interesting protein aside from all the structural proteins in the virion. That is the M2 protein. So here's influenza virus. It's an envelope virus surrounding the RNA genome. There are glycoproteins, integral membrane glycoproteins in the membrane of the virus. But there is also a very minor protein, between 4 and 16 per virion, called M2. And it's shown here. It's embedded in the lipid bilayer. This is actually an ion channel. And this has a role in entry of the virus into the cell, as we'll see next time. So again, another example of a specialized viral protein. Doesn't have a structural role, but serves some other function in the viral life cycle. Now, bacteriophages follow these rules of symmetry as well. This is an example of one of the tailed bacteriophages, the very famous ones you see in early images of viruses and some of the earliest ones studied. So the structure of this tailed virus was determined by X-ray crystallography and cryo-electron microscopy. You can see the head is built with the rules of icosahedral symmetry. This is a perfect icosahedron that has a T number. It's built with certain numbers of proteins that interact in quasi-equivalent ways. But you can see it also has some other components as well. So the nucleic acid is stored up here in this head, but it also has a neck, a contractile tail, and it has fibers that it uses to attach to the bacterium, and finally a base plate through which the DNA is injected. So when this phage sits down on a host cell, it pokes a hole into the membrane, using enzymes that are found in the base plate, and the DNA is injected through the tail at high pressure. But these are all components that are added to the icosahedral shell, which serve specific functions, just like the portal of herpes virus and the glue proteins of adenovirus. And finally, this is the latest structure of mimivirus, which is this huge virus that we've talked about. It's 750 nanometers in diameter. It is the biggest virus we know of. It has a 1.2 million base pair DNA genome, and the T number is 1,179. Remember, the T number times 60 gives you the number of protein subunits in the virion. These are huge. And I'm amazed that actually they were able to derive a cryo-electron micrograph structure of this virion. And here it is right here. You can see it's built just like an icosahedron. It's just really big. In the middle, this white material is believed to be the genome. You can see it doesn't have any order to it, but it does have density. And there's this very interesting star, which is completely asymmetric. It's only on one side of the virions. None of the other sides have this star. And the hypothesis is that this is what actually opens up when the nucleic acid gets out of the particle. So these blue lines represent areas in the capsid that can come apart and let the DNA out. And because they show up differently on the cryo-EM, that's why they're colored blue here, the interactions must be different amongst those proteins than amongst all the other proteins in the rest of the capsid. So another example of how you can have very specialized regions of even an icosahedral shell that allows you to do specialized functions. So the title of this is Complex Structures Are Challenging Viral Structural Biologists. So this is not easy to do because it's not only technically difficult to get the images, but the computational processes that are involved to solve the structures are quite large. And that's another example of how virology is attracting more people who are interested in computational aspects of structure.